My name is Ben Ellert, and I'm a research scientist in biogeochemistry with Agriculture and Agri-Food Canada. I grew up in a small mixed farm in southern Alberta here, and I've had most of my career here at the research center. We do work on carbon and nitrogen cycling, or biogeochemicals and the biogeochemical cycle. It's what makes the world go around. And I use a number of our long-term cropping system studies, some short-term, more agronomic studies, and the questions about land-atmosphere exchange of greenhouse gases never ceases. So I work with myself and a number of collaborators in that area. In agriculture, we are a little bit different than a lot of industries in that our greenhouse gases are biologically mediated. So in the business of agriculture is growing food and fiber, and that essentially depends on photosynthesis. Uptake of CO2 from the atmosphere by plants. So the land-atmosphere exchange of CO2 there's lots of interest in that. Everybody's interested in that we protect soil productivity by maintaining the organic materials that are there, and also interested in the prospects of accumulating soil carbon, which essentially pulls CO2 from the atmosphere, but also maintaining productivity, and the health of our soils depends on that organic matter. So that's the CO2 side. The other part is nitrous oxide. It gets much less press in most other industries, but it's a really major greenhouse gas from agriculture. And it's a small leak in nitrogen cycling, and the more nitrogen dynamics you have, generally speaking, you have more nitrous oxide leakage. So the chemical form of that is N2O. It's at about 300 parts per billion in the atmosphere, and it's been increasing since industrialization and agriculture use of nitrogen fertilizers especially, but any system with nitrogen cycling in it is going to leak N2O. So that's one of the things that we're looking at. Those emissions come from the soil generally. And the other big one is methane. And methane, the main source for methane emissions is enteric fermentation in the livestock industry. So we're concerned. And all these greenhouse gases, they act as sort of a small molecule audit of how efficient your agricultural system is. You have more leaking of methane from your cow to the atmosphere. That means less energy that you're putting into the cow and cow products, for example. And the same thing with N2O. If the more leakage of nitrogen, that means that's an indicator of doing a less well job of using that nitrogen to grow crops. So one of the main things I wanted to talk about today was some of the techniques we use to measure nitrous oxide fluxes from the soil with flux chamber, but also I wanted to talk about nitrogen fertilizer management and get an idea of how many people are using these or recognize these. So I brought a series of nitrogen fertilizer samples that you can look at and see what we're using. That, you can probably see it from 3 miles away. That's what fuels annual crop agriculture. It's urea. This is a polymer coated urea that slows down the release of that in the soil. This is a kind of urea, the trade name is Super U, and it is a sample of an enhanced efficiency nitrogen fertilizer is one of the things they call it. Why would it be enhanced efficiency? Does anybody use products like this? Has anybody heard of enhanced efficiency nitrogen fertilizer?
Yep.
Okay.
Coke industry promotes.
That's
If Riga says it's good, it must be good.
okay, and this is urea with Anvil. Anybody heard of that one? That's a relatively newer one. and that is another Coke product.
Is that the urease inhibitor?
Yes, yeah, that's a urease inhibitor. This is
2800 UAN.
irrigation producers like that one. That is 2800. The first number on your fertilizer is the nitrogen content. So these are mostly 4600. That's the percentage of nitrogen in the urea molecule. ESN has a little bit less. I believe it's 44 because there's the polymer coating and there's other stabilizers in there to maintain the integrity of the prills. I've also got a few non or less ammonical fertilizer samples some of you may be familiar with. This is 3400. What's the chemical form of that one?
Bomb.
Bomb. That's one of the that's one of the problems. It used to be widely used fertilizer, but because of its reactivity and use in explosives, there's lots of it going to tech mines in the Crow's Nest Pass for metallurgical mining, so we can all drive our Toyotas. But in agriculture, they have curtailed the use of that. For simplistic operational reasons, for some of our studies at the research center, we've taken to be using CAN 27. Anybody heard of that? Tom, of course. When I saw Tom here, I figured he's more qualified than I am to give this talk. This is calcium ammonium nitrate. So it's virtually the same form of nitrogen as 3400, only it's not just calcium, it's calcium carbonate and dolomitic limestone mixed with it. And when you do that, surprisingly, the thermodynamics of the substance change, and it's hard to make a bomb out of it. Okay, it's non-explosive. All of a sudden your transportation restrictions dissolve and you're fine. This is one of the things my agronomist friends really don't like me because I do crazy stuff. This is fertilizer used in the horticulture industry. It is calcium nitrate. Why would people be reluctant to use calcium nitrate in broadacre agriculture? Money plus, and the reason the money, you can tell, is the analysis. Instead of 4600, 15.500. It's a low analysis. And a lot of our producers, when you're trying to get the crop in the ground and you got a great big air seeder that was very foreign to me from my on the farm experience, that's a lot of material to handle, even when you're working with urea of 46% nitrogen. If you're working with calcium nitrate at a third that, you've got 3 times as much material to handle. So that's why it's not popular. For us to tease apart the reactions in the soil and that sort of thing, I think it's a very useful form of nitrogen fertilizer. So I like to use it in some of my studies, recognizing that it's not, there's not going to be a lot of uptake in commercial agricultural production. Why do I use that? Because urea is a very different chemical form than nitrate. And the differences between those two, how they feed the crop, I can infer stuff from that. Actually, I wanted Tom to get a look at this one because it doesn't look, it's supposed to be ammonium sulfate, but it's the most look like ammonium sulfate to me. I could be wrong. Tom will tell me.
I'll look at it.
Does anybody use ammonium sulfate? What might we use that for?
Canola.
Grass.
Yeah, it's ammonium sulfate is it's actually, I would watch for people trying to sell that to you. I understand it's a byproduct of nylon synthesis. So they've got a lot of it. It's widely used in Europe. It's a very good source of sulfate for crops that demand a lot of sulfate, like canola. In southern Alberta, I get more questions about what I say is sulfur toxicity. Salinity, our dominant salt here, is the sulfate salt, and we can have lots of sulfate in a lot of our soils. One other product, it's not a nitrogen product and it's not a pretty color, so I brought the actual product. This is, and I'm not endorsing any of these, but this is Entrench, and it is a liquid form of nitropyrin, which is a nitrification inhibitor. So you treat your urea fertilizer with this product and it slows nitrification. So those are an example of some of the different formulations of fertilizer that we're using to try to make better use of our nitrogen fertilizer in the system. The urease inhibitor, one of the most common one is this green color stuff. It's NBPT is the substance, and it slows the hydrolysis of the urea molecule. When you put urea, especially on the surface, the urease enzyme is everywhere. It's in the residues, it's in the plant tissue, it's everywhere. And it cleaves that ammonia, it cleaves the urea molecule, and it releases ammonia. Somebody mentioned the problem with ammonia volatilization. And even if you're banding fertilizer, that's one of the main reasons we want to band urea. We want to put it in the soil, and it's important to have good closure of that band of the soil slot that you cut, so that urea is buried and concentrated in a band. Otherwise, the urea molecules will cleave and you'll lose your nitrogen as ammonia, and that's not a good thing. It's money going up in the air, but ammonia is also an atmospheric pollutant. It's not maybe quite as bad as some of the other reactive forms of nitrogen, but it can definitely pollute waterways and go where you don't want it to. It's a loss of money, so we don't want to let that happen. And that's what the urease category of nitrification inhibitors are. Okay, so that inhibits that enzyme activity. The other transformation process that these so-called enhanced efficiency fertilizers target is nitrification. Nitrification is oxidation of ammonium ions to nitrate ions, okay? And generally speaking, in our cropland, we've got healthy, robust populations of chemosynthetic bacteria mainly. There's fungi that can promote that reaction as well, and they oxidize our ammonium to nitrate. Nitrate's a very important form of nitrogen for our annual crops to use, because our annual crops, compared to perennials, have a relatively paltry root system, and it can acquire nitrate. Nitrate is more mobile in the soil than ammonium. Why is that? Why is nitrate more mobile? There you go. Our soils have a cation exchange capacity, right? They absorb cations, positively charged things like ammonium. So ammonium doesn't move very far. When we were talking this morning about cover crops, we were worried about nitrate leaching. Nobody said anything about ammonium leaching. Why is that? I mean, ammonium can leach, but not as much as nitrate. Nitrate is mobile and it is prone to leaching, but it's also a good thing because crops can get it. But sometimes the key thing with managing your nitrogen, and it's easier said than done, is synchrony between nitrogen availability in your soil, what's in a nitrate form, especially for a nice canola crop like this, and the plant demands. When the plant is just germinating, it's just small. Nitrogen demands are small. When the plant gets to a stage like this crop wants nitrogen. Probably a week ago, nitrogen really started to peak for uptake on this. You can see a little bit of a response to nitrogen here. This is I would guess, a heavily nitrogen fertilized crop because it's nice and green, and you can see the ones that are a little bit lighter color are short on nitrogen.
But that one's 100, this one's 0.
100 and 0. There you go. See, I asked Jamie, I wanted plots that showed a nitrogen response, and they deliver. What's not to like about that? So these are some of the ways that is being proposed, and I'm not sure of all the programs, Ken, but there is talk to one of the things that they are considering is programs to encourage the adoption of these enhanced efficiency fertilizers. Okay, and what that does, the idea there is to make better use of that nitrogen fertilizer to delay its availability in the soil. And I know if you've been at previous field schools, I'm sure I heard Tom Jensen talk about this a few years ago. So it's something that we've been looking at. I guess right now there is a new sort of imperative to take a closer look because they want to cut back on nitrous oxide emissions. One of the big things that, from my perspective, that's cutting back on nitrous oxide emissions is the expense of nitrogen fertilizer. It's one of the most expensive inputs to agriculture on an annual basis, and it's one of the most energy intensive inputs. It's not just the nitrogen that we're using and the nitrous oxide leakage, but when we do accounting of the greenhouse gases from agriculture, we have to account for the fossil fuels it's consumed to manufacture that nitrogen fertilizer. The triple bond, we're swimming in nitrogen, lots in the atmosphere. 78% of our atmosphere is N2. It's not available to most plants except those that are capable of biological end fixation. So that's why there's interest in pulse crops, in soybeans, especially alfalfa, and these kinds of things that can fix nitrogen. Soybeans, those kinds of things. But for our crops like canola and cereals, agriculture is not a perpetual motion machine. We have to recycle that nitrogen. And these are some of the products that we use to manage that. Another form we talked a little bit about, and maybe some of you can help me on this. I was grasping at straws. This is a sample of feedlot manure. You probably don't recognize it as such because it's just before, it needs one other step of grinding before we analyze this in the lab. Manure is really variable concentration of elements, but with urea that's 4600, the numbers I put on this, and you can correct me if I'm way out to lunch here. 1.7% nitrogen, 1.2% P2O5, 2.1% K2O, and 0.2% sulfur.
Right on.
It's hugely variable, the composition. Those are just representative numbers. Not too bad, Tom?
It's
okay.
A lot of manures that close to that.
Close in there. So that's the thing that we're talking about. Livestock production system, it's all great to have circular agriculture, and there's lots of things that we can do to manage those nitrogen. There's lots of leakage prone to this. I've got colleagues that have spent a career on trying to make effective use of this. But that's one of the things as a nitrogen source. I've got this at 1.7% N. I said that producers would never use calcium nitrate because it's 15.5%. This has a relatively low nitrogen percentage, and to make matters worse this is dry basis. That's a percentage on a dry basis. In reality, when you're handling manure, a quarter to a third of it is dry matter and the remainder is water. So we've heard lots about composting. Composting is mainly a way, it probably stabilizes the nitrogen a little bit, but it's mainly a way to dry it out so you can afford to move your nutrients farther from the confined feeding operations. So that's a little bit on managing manure nitrogen. This morning I've got to check my friend and colleague Kevin Float's Cow Patty Critters book about the nitrogen dynamics. I don't know if Scott's still here, The nitrogen in a cow, as I understand it, it's really partitioned between urine and feces, and the lability of that nitrogen differs quite a bit. There's lots of nitrogen that's excreted as urine, and that's very important in grazing systems. So when you go to places that are pastoral, like New Zealand, they are very concerned about losses of nitrogenous gases from urine patches. Less so from the fecal patches because it's largely organic nitrogen and it's more stable. A huge difference in the chemistry between those. Manure is really complicated because it's a hodgepodge of everything. This is a feedlot manure, and I had to specify it wasn't composted, it was stockpiled. It's from concrete apron by the feed bunks, and the bedding in the pens is straw. All of those factors influence what manure is, and manure isn't the same as urine or feces or bedding. It's a hodgepodge of all those. That's the nitrogen that we have to work with. How am I doing time-wise? I've got everybody just in. So one of the things that there's lots of interest in, and the phone never stops ringing, about trying to measure nitrous oxide fluxes. Okay, and to do that, it's relatively simple. We use a chamber, we push it into the soil. There's also chambers, a little bit different configuration. Actually, the ones in this plot are rectangular. They've got a rectangular footprint, so they're ideal for measuring fluxes from a system where you've got fertilizer bands. But this is a flux chamber. It's got a seal there. It's vented to the atmosphere for aerodynamic reasons. And this is we collect the samples in the field. We use a syringe with a needle on the end. We have glass vials that's got a rubber septa in them, and they're evacuated, so they're under vacuum. We take a sample of chamber headspace gas with a syringe, and we transfer it to the vial. So we put, at standard temperature and pressure, we put about twice as much air or headspace gas in this vial as what the vial volume, water volume is. So it's overpressure by about 2 atmospheres. Typically we'll take a sample at 0, 15, 30, and 60 minutes. Okay? And we get a change in the concentration of greenhouse gases in the headspace of that flux chamber. Okay? About one of the most straightforward ones is nitrous oxide. Nitrous oxide concentration starts at the ambient level, about 300 ppb. That's parts per billion. It's low. We have to use sophisticated instrumentation in the lab to analyze that. 300, the next one is a little bit higher, next one's a little bit higher, next one's a little bit higher again. And we fit a curve and we estimate the flux for that 60-minute deployment period of N2O emissions, the transfer of N2O from the soil to the atmosphere. That N2O has a very long atmospheric lifetime, and each N2O molecule is about like 300 CO2 molecules. So it is a potent greenhouse gas. That's why we're concerned about it. Okay? So that's how we measure nitrous oxide flux. At the same time, we are also measuring methane concentrations in that gas sample. What happens to methane concentrations in my flux chambers? Any ideas? Oh, come on.
Over time?
It decreases. Tom knows. So our soils actually scrub methane out of the atmosphere. Okay, all of my friends, and that's why I'm careful with the cow people in my lab, because I want to quantify, we're starting at ambient methane, which is about 1.7, 1.8 ppm of methane, and it decreases. Why? Because the soil microbes are so hungry for carbon, they take that methane and they oxidize it to CO2 as an energy source. So our soils scrub methane out of the atmosphere. Unfortunately, they don't scrub near as much methane out of the atmosphere as we need to do to take care of what our bovine friends are doing. Bovines are emitting lots of methane. And another source of methane that colleagues and ag engineers are working at is anytime you have an anaerobic manure storage facility, you have potential for methane emissions to the atmosphere. So we measure that. What about carbon dioxide? What happens to carbon dioxide in my chamber? Soils emit CO2. So it's like N2O. Put the chamber on, we will measure, and that's the thing, if you don't believe in the changing, or maybe it's a sign that I've been in this game for too long. When I first came to Lethbridge, I would calibrate my analyzer to measure CO2 in the field, and if it was measuring about 360 parts per million of CO2, good, it's working. Now if I come out here and it's measuring 360 ppm, it's wrong, because we're at about 410, 415 ppm of CO2. And it does vary. This is biogeochemistry. We've got seasonal variations in our atmospheric CO2 concentration. It's not the same every place on the planet, and especially in the northern hemisphere where we have seasonality. In the summertime, all of our lush vegetation pulls down CO2 concentrations. In the wintertime, it increases. So we've got these diurnal fluctuations. And using biogeochemistry, we can also tell you whether the changes are due to fossil fuels or whether they're due to natural vegetation. Those are the kinds of things we can do. One of the things that I get a little bit, and right now I'm doing my best to educate managers and policymakers and that sort of thing, is they like to speak as though N2O flux, soil N2O fluxes are something completely separate and different from CO2 fluxes. And then I get people and they get all worried about why my flux chamber has no vegetation in it, okay? In reality, the land atmosphere exchange of N2O and CO2, they're very analogous kinds of things. They both matter. In some ways, nitrous oxide emissions are simpler because it's the soil microbiota that generate the N2O, and then it leaks to the soil surface and eventually the atmosphere. CO2 is much more complicated in a growing system, and we can measure soil CO2 emissions, but if I've got a plant in my chamber and I put a lid like this over top, what's going to happen? Plant physiology, first year biology. Plants are photosynthesizers. For them to photosynthesize, they need photons. And it is amazing, we can measure this. You put a cover like that over top of a plant and it changes from taking CO2 out of the atmosphere. There's no more photons anymore. It emits CO2. So for measuring greenhouse gas fluxes using this static chamber approach, we like to use a vegetation-free surface area. And I think my colleagues, I don't know who's doing the greenhouse gas measurements in here, they're mostly picked free of vegetation. Usually when we do a flux, we go in and we pick all the weeds, because that does influence it. But really, they talk a lot about soil, I talk a lot about soil carbon sequestration, the land atmosphere exchange of CO2, and that entails measuring fluxes. The difference between measuring soil carbon sequestration and N2O fluxes is the time scale. For measuring soil carbon sequestration, I like to measure fluxes on a time scale of 5 years. I'll do a baseline sampling, And very carefully, I'll come back 5 years later and I'll do another sampling, and that gives me my net exchange of CO2 between soils and the atmosphere. I don't have the luxury of doing that with nitrous oxide. Nitrous oxide comes off in fits and starts. And it's very expensive to measure nitrous oxide because one flux measurement isn't enough. I need at least 25 flux measurements a year to you need a flux measurement campaign to measure the annual transfer of nitrogen from the soil to the atmosphere in the form of N2O. Okay, and that's important from a greenhouse gas perspective. Out here, sort of our, well, not the worst case scenario, but a site that's leaking a lot of N2O, irrigated system, heavy N fertilization rate, you might have 3 to 5 kilograms of N2O emitted from the soil to the atmosphere in a year. 3 to 5 kilograms of nitrogen. That's not a huge amount, frankly. Environmentally, it's very impactful. Nitrous oxide is a potent greenhouse gas. Oh, and because it's long lived in the atmosphere, it eventually finds its way to the stratosphere and it catalyzes ozone destruction. So you've got a double environmental whammy from nitrous oxide. But for agriculture, what I'm trying to sell is we should be doing a better job of measuring nitrogen fertilizer recovery. And that's an age-old, long-term challenge that we've been facing in agriculture. Tom Jensen's done some very good work on that, and I learn from him all the time. We are going back in with some of these enhanced efficiency fertilizers, and we're using a little bit of a different labeling technique. It's an isotopic technique, but it's very different. We're using a low-level technique where we're fertilizing entire small plots. Plot this size, the whole thing gets labeled. And instead of using 2 atom percent, we're using 0.1. At least Tom will know what that means. We're using low atom percent. And the thing that we can do is, I think we can do a better job of measuring how efficiently our nitrogen fertilizer, our crop is using it. We're letting our crop define what is available from this fertilizer. You go to the literature and you see these studies, looking in the Canadian Journal of Soil Science, and they've got nitrogen fertilizer use efficiency. They've got data that range from 20% up to 70%. The p-values are not significant. You can't measure it. It's messy in a field plot study. We found that with the N15 technique, we can improve that by an order of magnitude. Okay, so we can really resolve our fertilizer end use efficiency. Plus, for the producers, I'm excited about this approach because I think it matters to the producers. It's the whole, it doesn't just measure N2O loss, it measures all the losses. This morning when we were talking about cover crops, they mentioned leakage in gaseous form, nitrogen as N2O, but also nitrification. A lot of it leaks as N2. And that is very challenging because we're measuring a little leak of nitrogen into an atmosphere that contains 78% N2. Very hard to measure that, but we can measure it with this. We measure all of our losses. Other gaseous losses, of course, are ammonia. You can lose ammonia molecules. So if you have a urease inhibitor like Anvol, Anvol is just NBPT. It's like agrotane, only this has another chemical in it that eventually breaks down into NBPT. So it's a longer lasting, it's supposed to be. I haven't worked with it yet. But that's what we can do with our isotope techniques. Got a question over here.
Dr. Ellert, is there any differences in the nitrogen loss mechanisms in manure towards N2O as relative to urea?
Manure versus urea?
Yep.
Yes, there's huge differences.
Worse or better?
Oh, it's different. Manure systems they're challenging. I think it's tough to measure N2O leakage and gaseous leaks from our crop systems with nitrogen fertilizer. Manure is even worse. One of the things that I find, and I have done work on our irrigated systems with manure, what you get with manure application is you're putting on carbon as well. I didn't give you the carbon number on this. This contains decomposable carbon as well. And at microsites, what happens is that carbon decomposes. And when the carbon decomposes, it locally pulls down your O2 concentration, and then that's when you have propensity for nitrogen reactions. It makes a lot of N2O. So you're prone to that with manure, okay? And some of these same technologies, especially the urease inhibitors, I think there's directions for N-trench. You can put that in liquid manure systems, and I don't know if there's a lot of work done on that yet. I'm not that good at that. I probably shouldn't have brought that fly in the ointment to this discussion, but yeah. I find it kind of intriguing. Our friends in New Zealand, and this goes back a ways, they were going to address their greenhouse gas problem. Lots of ruminant livestock production, lots of dairy in New Zealand. The urine patch is a huge issue in New Zealand. And they were going to solve all their problems by putting DCD everywhere. So that's the same nitrification inhibitor as in here. And they were going to stop all their leaching of nitrate, and everything was going to be lovely. And they've got all kinds of ingenious ways to track where the cow's urinating and where you put these inhibitors on. And at one time they were even feeding DCD to the cows. And that was all fun until the Chinese, who import a lot of that milk product, dairy product, they found DCD in their products. And given all the melamine scare and things that happened earlier, that put an end to that whole idea. So I'm a little bit reluctant and concerned about ever more use of synthetic chemicals, of which these are in our agricultural systems. But it's something that is on the agenda right now, so we're studying this. And I have colleagues doing a lot of N2O measurements with these so-called enhanced efficiency products. So there's literature out there on that. And we're measuring total fertilizer recovery by the crop with these as well. And we hope to get some more funding to do that. One of the other things, Ken mentioned genetics, plant genetics. And one of the other things that you may be thinking about, I know colleagues, plant geneticists, are looking at this, and it's a concept called biological nitrification inhibition, okay? And they have studied mainly perennial crops, grass crops, certain grasses, and they're honing in on the phytochemicals that do the same thing to nitrification that these synthetic chemicals do. And there are people right now looking at that as a breeding target to try to introduce those into annual crops. Whether that's a feasible process or not, I am not sure. When you look from an evolutionary standpoint, you've got a perennial grass that's there. It's had a year or 3 or 30 to amass a perennial root system that persists from one year to the next. That kind of a plant can afford to inhibit nitrification. Why? It's got a huge root system, and if the available form of nitrogen is ammonium, it's got a root right in the neighborhood and it can get it. An annual crop, our annual crops feed predominantly on nitrate because nitrate is more mobile. So whether that's going to work out, but that's something that you may be hearing about in the next 10 years. I don't know. Biological nitrification inhibition. That's what that concept is. Any other questions or comments on nitrogen fertilizer management, greenhouse gas exchange? Tom.
I know on the federal program, the goal is to reduce nitrous-oxide emissions by 30%. Initially, that wasn't communicated because people just heard reduce nitrogen fertilizer use by 30%. Any comment on?
I'd like to defer to Ken since he's the politician amongst us here. Where, but yeah, you know, everybody is different on their perspective. You know, I was inquired early on about this, and maybe I'm more of a pessimist, but I said this isn't possible. This is unrealistic, and it all depends on what you're talking about. With these flux chambers, we are measuring what we call direct N2O emissions, okay? And that is the N2O that's emitted from the soil pores there into my flux chamber. And that's the direct N2O emissions. And there's a whole other category, probably less, maybe another 25%, I hope not more, it could be more, that we call indirect N2O emissions. And we've got even precious fewer data on those, okay? There's very little data. What indirect N2O emissions are, that's when you have a beef feedlot that's spewing ammonia into the atmosphere like crazy, no offense. They don't have to put manure on the field because they got enough atmospheric deposition of urea from that barn right there. Nobody counts for that, but ammonia is particularly bad because it goes everywhere. It goes into Jail Lake, for example. And in Jail Lake, I've never measured it, but I would be really surprised if that's not a big N2O source to the atmosphere. And there's N2O leakage from landfills and all these kinds of things. Generally speaking, no. When you have an environment such as a landfill or a manure storage or that sort of thing that is emitting methane, that means chemically the system is too reduced for N2O to be stable, so your nitrogen is not leaked as N2O, it's reduced all the way to dinitrogen. The farmer, I think, still should be concerned about that because that's a loss of nitrogen that you're going to pay for and replace next year. But environmentally, it's not near as bad as N2O. So that's the other thing. Yes, I was talking to one of my counterparts in industry and I was surprised that he wasn't aware of this. Nitrous oxide, everybody makes a connection between nitrous oxide leakage and what process? Gerber. Denitrification. And that's true, that's right, but it's only half of it. The other transformation that contributes to N2O emissions is, I heard it,
Nitrification.
exactamo. That's the much neglected one, and that's one of the things that I've done some research and spent your tax dollars on, and I wish I had a better explanation. But for managing soil N2O emissions, it would be really nice to know whether that N2O is being formed by denitrification process where there is low oxygen availability or whether it is formed by nitrification processes. And I learned strategies and approaches that aren't any good to do that with. But we're still working on that. We don't have a good handle for the relative, we can guess, but we don't have a really good feeling for the relative importance. But we believe that at least half of our N2O emissions from the soil are associated with nitrification, not denitrification. And that sort of we allude to, some years we sure wish we had a nitrate leaching problem. In this part of the world, nitrification is a very important process for N2O leakage.
Just a question. That's when the ammonium is getting changed to nitrate? Is it? Or is the nitrate?
It's everything. There's lots of points in that dynamics. There's a lot of advances being made in the soil microbiome. so they can probe individual enzyme systems that catalyze these reactions. And every time they look at it, they discover a previous pathway that people didn't fully appreciate before. So the biochemistry is actually quite involved. That gives me a little bit of a segue. I don't know if anybody has heard of ammonium thiosulfate. Why do we not want to use ammonium thiosulfate? In my opinion, from a nitrous oxide perspective, it blocks the nitrite to nitrate transformation. And whenever you get a buildup of nitrite in your soil, in my experience, that's when you get N2O production and eventual leakage. All was not lost on my research to try to disentangle the relative contributions of denitrification and nitrification to N2O production in the soil. One of the other things that I really did gain a good appreciation for is when we're looking at N2O in the soil, everybody talks about what are the processes that make N2O. Where is that coming from? How can we stop that? And from that work where we're trying to understand this, one of the things that we realize that nitrous oxide consumption in the soil is a very important process. So we found that in a heavily irrigated system where we had lots of nitrate due to improper management, actually, a lot of times when you terminate that alfalfa stand and all those fine roots start to decompose, you can get nitrate at depth. We follow that and we had quite a bit of N2O there, but as that N2O diffused through the drier layers of the soil, it was reduced all the way to N2. So it was much less of a problem than it could have been had N2O consumption in the soil not happened. So I'm sorry if none of this isn't practical enough for you. I hope it's at least of interest to give you an idea of some of the things that we're concerned about. One thing that is practical is I'm advocating for direct measurement of total fertilizer N uptake by the crop, and maybe not putting all of our emphasis on the very expensive and laborious N2O measurements.
We got one question over here.
I was going to ask, like, for your CO2 measurements, is there a difference in crop growth with a higher CO2 concentration?
A difference in?
Crop growth. Like, if you have say you have a system that's 300 parts per million, and you have a system that's 400, does the crop grow better or worse, or is there no difference?
That depends a lot, and generally, in most circumstances, there is an increase, especially with C3 crops like our wheat and barley and canola and that sort of thing. If you have a higher CO2 concentration, that boosts your yields a little bit. The balance between photosynthetic uptake and photorespiration is a little bit in favor of greater net primary production. There's been lots of work done on that, especially in natural ecosystems, grasslands and forests, because it's not just our agricultural land, that's only a portion of the terrestrial surface area. There's lots of other ecosystems, and we're trying to foresee how these other ecosystems might behave and respond to changing environmental conditions. I find it really interesting in southern Alberta. Our tomatoes are grown in greenhouses at Medicine Hat, and Medicine Hat happens to be on a very interesting gas field that is very depleted in carbon-13. So when I analyze, again, this is an isotope thing, some of it, and they are very depleted in carbon-13 because those tomatoes are grown in the wintertime in an atmosphere with elevated CO2 because productivity is more. You'd never see that kind of C13 signature on a field-grown tomato. Well, unless you had a free air CO2 enrichment study. But yeah, CO2 matters. It's not going to solve all your problems. Higher CO2 levels, at least in C3 crops, tend to close the stomates a little bit. You get greater water use efficiency. Because because they don't need to keep the stomates open as large to get the CO2 that they need. So corn less so. Less benefit for a C4 crop like corn, because it's already designed, corn evolved at a time in Earth's history when atmospheric CO2 levels were very low, and it's got certain physiological tricks to concentrate the CO2 in a different part of the stem. All fascinating plant biochemistry.
Well, I usually like to say that I've always enjoyed Ben's presentations, but he called me a politician today. So I think it's really important that we understand the system, that we can be able to best employ practical approaches to hit the goals that we're doing. And yes, the government actually did say fertilizer at first, the 30%, and their communications team all got fired. But that is the goal in the European Union. It is 30% fertilizer reduction. And that's what kind of got everybody in Canada all riled up about, okay, the government's going to tell me how much I can fertilize. That's not good. However, it just so happens that not even knowing about it, you're standing behind a plot that demonstrates, at least visually, that maybe we can look at fertilizer reduction. The plot right there is 100%, looks nice and green. Beside it is the check with nothing. This plot right here is 75% and it looks as good as the 100% chance. So a lot of, fancy chemical salesmen will tell you that, yeah, you can reduce your fertilizer and then buy my product and you'll have the same yield. I would encourage you, when you're doing that, to try it without the product they're selling you. And we have seen this a lot in our own research, is that we've kind of improved our soils, our cropping systems with the adoption of zero tillage and better crop rotations and all that, that a lot of times we actually can reduce our fertilizers without seeing a yield hit. The problem is we use nitrogen as a risk management tool. What if I happen to have that perfect year where I need that extra and I can get that 1 year out of 10 that's a bumper crop? So the practical side of this is being able to tweak out and understand with the right data and the right knowledge where you can tweak your systems to improve your nitrogen use efficiencies. We talked about legumes. Like, I'm not afraid of hitting 30%. You can just grow 30% legume crops, and you've done it. So politically speaking, the folks that are already doing all of these things, they're the ones that are a little bit annoyed, but there are some laggards out there in the industry as well. So I appreciate understanding. I've learned a number of things today as well. Understanding the cycling and the challenges as it relates to greenhouse gases. And I'd like you to join me in thanking Ben for another interesting talk.